Glycosylamines, a sugar in which an amino group replaces the anomeric hydroxyl group, is called a glycosylamine. Examples are beta-D glucopyranosylamine and adenosine. So this is the structure of beta-D glucopyranosylamine, where this NH2 is actually replacing the anomeric hydroxyl group, and this is the structure of adenosine, where this group is replacing the anomeric hydroxyl group. Nucleosides are glycosylamines in which the amino compound or the amino component is a pyrimidine or a purine base or something like that, and in which the sugar component is either D-ribose or 2-deoxy-D-ribose. That means it may be D-ribose or it may be deoxyribose. Amino sugars, a sugar in which an amino group replaces a non-anomeric OH group, is called an amino sugar. So if the amino group replaces the anomeric OH group, that is called glycosylamine, and if it replaces a non-anomeric OH group, then it is called amino sugar. This is the structures of amino sugars. D-glucosamine is an example of an amino sugar. This is the beta-D glucosamine, where this is the anomeric hydroxyl group, and this amino group has replaced the non-anomeric hydroxyl group. In many instances, the amino group is acetylated as in N-acetyl D-glucosamine. This is the N-acetyl D-glucosamine, where this amino group has been acetylated. N-acetyl muramic acid is an important component of bacterial cell walls. This is N-acetyl muramic acid, where this R group is this one. D-glucosamine can be obtained by hydrolysis of chitin, which is a polysaccharide found in the cells of lobsters and crabs, and in the external skeletons of insects and spiders. So this is the polysaccharide chitin, and you can obtain D-glucosamine if you hydrolyze this. If you just break it, you will get the D-glucosamine, and this also will be broken. N-acetyl D-glucosamine is liberated when chitin is hydrolyzed in aqueous acid. Stronger acid brings about hydrolysis of the amide bond to give D-glucosamine hydrochloric acid and acetic acid. So you have taken the chitin, then you are just hydrolyzing it with strong acid, then you will get N-acetyl D-glucosamine, this one, and if you concentrate the acidic strength, then you will get D-glucosamine with the HCl salt. If you apply this part will also be hydrolyzed. Glycolipids and glycoproteins. Carbohydrates joined through glycosidic linkages to lipids and to proteins called glycolipids and glycoproteins, respectively. These control the entire activities in the cell. Most proteins are glycoproteins, of which the carbohydrate content can vary from less than 1% to greater than 90%. Tumor cells also have specific carbohydrate markers on their surface as well, a fact that may make it possible to develop vaccines against cancer. The human blood groups offer another example of how carbohydrates in the form of glycolipids and glycoproteins act as biochemical markers. The A, B, and O blood types are determined respectively by the specific A, B, and H determinants on the blood cell surface. The A, B, and H antigens differ only in the monosaccharide units at their non-reducing ends. The type H antigen is the precursor oligosaccharide of the type A and B antigens. So this is the structure of type A determinant, type B determinant, and type H determinant. I don't know much about these things because these are the part of biology, and actually I don't know much more things about biology. So if you are interested, then you can just search about these things. Carbohydrate antibiotics. One of the important discoveries in carbohydrate chemistry was the isolation of the carbohydrate antibiotic called streptomycin in 1944. Streptomycin disrupts bacterial protein synthesis. So streptomycin, the work of streptomycin is to disrupt the bacterial protein synthesis. Now this is the structure of streptomycin. These are the structural units. Unit 1, this one is unit 1, this one 2, this one 3. Other members of this family are antibiotics called kanamycins, neomycins, and gentamicins.